Welcome to all NEET aspirants. Today the first revision of biotechnology, the concepts, the interlinked aspects in form of MCQs, statements, the true or false with regard to biotechnology. With regard to this first question, write answers to the following questions with regard to the activity of E-core 1 restriction enzyme on particular nucleotide sequence. The E-core 1 which act on GAA TTC. Phi3 CTT AAG 35, and this is the palindromic sequence. It reads same in both forward and backward direction. GAA TTC, GAA TTC. So in between guanine and cytosine 3 hydrogen bond 2. So total number of hydrogen bonds 1, 2, 3, 4. 4 into 2 8 plus 3 11 plus 3. Total it is 14 hydrogen bonds. And E-core 1 breaks between G and A. It breaks between G and A. So and here the two fragments which are formed, it is single stranded. At the end of or it is double stranded, two fragments which are formed. So in this adenine guanine which are the purines, so 1, 2, 3, 4, 5, 6 GAA and GAA which are the purines. So it is 6 purines, 6 pyrimidines. Total it is 12 nitrogen bases. And purines which are dicyclic, pyrimidines which are monocyclic. In case of purines, it is dicyclic and which are monocyclic. With regard to the questions, number of phosphodiester bond it breaks. It breaks 1, here 1. Number of phosphodiester bond it breaks, it is the 2. Length of palindrome 1, 2, 3, 4, 5, 6. Total number of nitrogen bases it is the 12. For 20 nitrogen bases 34 angstroms. For 12 nitrogen bases 12 into 34 by 20. It comes under the 20 point, the 5 angstroms something. Length of the DNA is 20.5. And with regard to this what it is, number of double bonds disrupted 1, 2, 3, 4. Adenine, adenine, thymine, thymine 1, 2, 3, 4. The 4 double bonds disrupted. Here it is 20 point something. Length of palindrome, that is angstroms. Number of hydrogen bond intact, guanine cytosine 3, cytosine guanine 3. So number of hydrogen bond intact it is 6 hydrogen bond intact. Number of triple bonds present, it is 1 triple bond, it is 1 triple bond. 2 triple bonds which are present. Total number of hydrogen bond in that palindrome 3 plus 2 plus 2 plus 2 plus 2 plus 3, it is the 14 hydrogen bond in that palindrome. 14. Total number of carbon nitrogen rings in that palindrome. The purines which are 6, pyrimidines which are 6. Purines 2 carbon nitrogen rings, 6 into 2, the 12. Pyrimidines having 1 carbon nitrogen ring, 6 into 1, 6. It is 18 carbon nitrogen rings. So, in the simple, we elaborated in the form of varieties questions for this ecore 1 act on particular nucleotide sequence, GIA TTC. In this question, with activity of ecore 1 in above sequence in a linear DNA, these are the questions. How many fragments formed? Number of fragments having single stranded ends at one side of double stranded DNA. C. Number of fragments having single stranded end on either side of double stranded DNA. D. If above sequence, this sequence circular, how many fragments are formed? These are the questions. The ecore 1 will act between G and A. G and A, and the phosphodiester bond, as it breaks, so it is single strand and it is double strand. At the end of the double strand, single stranded fragment it is. It is the single strand and it is the double strand. And it is double strand, it is single strand, double strand, it is single strand. It is double strand, it is single strand. So, it is double strand, it is the single strand. So, your number of fragments 1, 2, 3, 4, 5. So, number of cleavage points it is the 4, number of fragments it is the 5. It breaks at 4 points, number of fragments we got 1, 2, 3, 4, 5. If it is a linear, the number of cleavage points are 4, the final it is n plus 1 in case of linear. 4 cleavage sites plus 1, we got what it is 5 fragments. How many fragments formed? 5 fragments formed. Number of fragments having single stranded on one end of double strand DNA. Single strand on one end of double strand. This is 1. This is 1. How many it is? The 2. Number of fragments having single stranded end on one side of double strand DNA. It is double strand DNA, it is single stranded sticky end. Number of fragments having single stranded on either side of double strand DNA. It is a single strand, it is the double strand. Single strand, double strand. Single strand, double strand. How many fragments you got? 1, 2, 3. Number of fragments having single stranded end on either side of the DNA is the 3. Total it is 5. If above sequence it is circular, how many fragments are formed? If it is a circular, the principle it is n is equal to n. Number of cleavage points is equal to number of fragments. Number of cleavage point restriction site, the 4. 4 are found. All these 4 fragments are like this only. Single strand on either side of what it is double strand. No fragment is going to be like this. Every fragment is going to be like this in case of this sequence. And very important in case of the sequence point of view. So keep watching our video lessons for further number of questions. Thank you very much.